Good morning, everyone. The name of our lecture is Physiology of Vestibular Analysis, Test, and Olfactory Analysis. What we are going to discuss today? We are going to discuss anatomy and physiology of the vestibular apparatus, physiological mechanism of the equilibrium, sensation of taste and its physiological mechanism, physiology of olfaction, olfactory signal and its transduction. First of all, let us begin with vestibular analysis. So, anatomy of vestibular apparatus. Vestibular apparatus consists of two parts. Its utricle and suckle, which provide information in both linear acceleration. Also, they're responsible for static, in other words, for static acceleration. And three semicircular canals anterior, posterior, and lateral, which detect rotational acceleration or dynamic acceleration. Each utricle and suckle have the macula with the hair cells. Hair cells will project into endolymph, where hair cells are embedded into gelatinous autolytic membrane. Autolytic membrane consists of calcium ions that resist change in movement. Utricle more sensitive to horizontal acceleration. During forward acceleration, autolytic membrane lags behind hair cells, so hair cells will push backward. Suckle more sensitive for vertical acceleration. Hairs pushed upward when person is distant. So, macula is that structure which is common as for succula as for utricle, which consists of the hair cells. Hair cells, they are embedded on that autolytic membrane, and in autolytic, as we discussed, hair cells consist of the stereocilia and the kinocilium, and the movement of hair cells will be in the direction of the acceleration. So hair cells will move due to the movement of the autolytic membrane. So autolytic membrane will push these hair cells to the direction of acceleration. Each hair cell will be innervated by the vestibular nerve. It is the 8th cranial nerve, which will send information to the higher centers of the nerve system. So, the detections of the macula of utricle and suckle is going to detect ventricle and horizontal acceleration, and as a result, in this situation, autolytes which are located upper of the autolytic membrane are going to send signals to the autolytic membrane, which will push hair cells on the direction of the acceleration. In the structure of the hair cells, the stereocilia is going to bind to the kinocilium and as a result produce a depolarization, which is going to summate the package of action potential are going to summate and send information to the 8th cranial nerve. So let us discuss about the hair cells more in detail. Hair cells, their receptors, hair cells receptors have stereocilia and kinocilium. When stereocilia bend toward to kinocilium, membranes depolarize and release neurotransmitter that stimulates dendrites of vestibular part of 8th cranial nerve. When they bend away from kinocilium, hyperpolarization occurs and the action potential will not be spread. Frequency of action potential carries information about the movement. So we have here kinocilium, which is going to bind to the stereocilium. First of all, we'll open the potassium voltage-gated ion channels, which are going to cause opening of calcium ion channels. Calcium ion channels will be responsible for movement of calcium ions inside of the stereocilia and as a result responsible for production of depolarization. Due to the situation, depolarization, the cells are going to release neurotransmitter, which will stimulate dendrites of 8th cranial nerve and produce excitation and spread. The excitation. Function of the semicircular canals. As we discussed, we have three parts of the semicircular canals. Duct is anterior, posterior, and lateral, and they're responsible for for acceleration in a different position. So it could be in anterior, it could be anterior acceleration, posterior acceleration, and also lateral in case when you will move head in one shoulder and another shoulder. And due to this kind of acceleration is responsible for rotation. So the situation will be the similar, where hair cells which are embedded to the autolytic membrane, they are going to push on the direction of the acceleration due to the movement of the autolytic membrane. So, semicircular canals provide about rotational acceleration. So they project in 3 different planes. Each canal contains semicircular duct. At the base of this semicircular duct is located crista ampullaris, where sensory hair cells are located. Hair cells processes are embedded in the capula. Endolymph provides interior so that the sensory process will bend in the direction opposite to the angular acceleration. So you could see here where endolymph in semicircular canals, then copula hair cells, and due to the rotation moment, during the movement, endolymph will bend to the copula and in opposite direction to the angular acceleration. What about the vestibular tract? So information from the 8th cranial nerve, from the vestibular cochlear nerve, will be spread to the, first of all, to the level of the medulla oblongata, where it will bind to the vestibular nuclei. After that, information is going to spread to the cerebellum. So in this case, information about the equilibrium will be sent to the cerebellum, and cerebellum in this way will control and control the muscle contraction, and also it will be responsible for contraction of the muscles during the movement and to maintain the equilibrium. Also, the nuclear, also information is going to spread through the thalamus to the vestibular cortex, which is responsible for awareness of spatial orientation and movement. Also, when we talk about that some tract from the medulla oblongata will be spread to the nuclei of eye movement, mostly it's located to the corpora quadrigemina superior colliculi, which is responsible for oriental reflexes. In this case, when we talk about oriental reflections, when you will see something or you are concentrating on something, the head will rotate on that the direction which you're interested in. Physiological mechanism of equilibrium. Equilibrium of the body is reached through the interpretation of the response of the vestibular receptors, visual input, stretch receptors, which are located in the tendons, ligaments, and muscles, input from the muscles and tendons. 2 types of balance which we discussed of equilibrium are static equilibrium, which will be done by the function of the cyclic and utricle, and dynamic equilibrium, which will be done by the function of the semicircular channel. So information from the eye, from the vestibular apparatus, from the joint tendons, muscles, together will be spread to the vestibular nuclei in the brainstem. This information will be spread to the cerebellum, and as a result, cerebellum, like the part of the nervous system which is responsible for control of the muscle movement, and also it is responsible for not only for controlling of muscle movement, but also it has its own cerebral spinal tract, which is responsible for muscle contraction, will affect on the effector organs. In our case, it will be skeletal muscles, and this way it will coordinate the muscle movement. Also, the information from the vestibular nuclei will be spread to the oculomotor central, which is responsible for control of the eye movement, and in this case, eye will move in the different directions, and also during the rotation of the head. and information will be spread to the spinal cord for controlling of the body movement. What is a nystagmus? Nystagmus and its clinical significance. Nystagmus is involuntary oscillation of the eyes when spin is stopped. Eye continues to move in direction opposite to the spin, then jerk rapidly back to the middle line. When person spins, the bending of cupula occurs in the opposite direction. As the spin continues, the cupula straightens. Endoleaf and cupula moving in the same direction as speed, and speed affects muscular control of the eye and the body. If movement suddenly stops, the interior of endoleaf causes it to continue moving, but in the direction of the spin. Pathological nystagmus occurs without spinning, and it informs about the central nervous system disorders. So you could see here the movement of the eyes during the when the spin is going to stop and how it's going to happen. Okay, now let us continue and discuss about the sensation of taste. The sensory structure that detects the taste stimuli are the taste buds. They are oval structure embedded in the epithelium of the tongue and the mouth. Most taste buds are located in the epithelium of the papilla, which is small. Right areas of the surface of the tongue. Taste buds are also located on the areas of the tongue, the palate, and even in the lips of the throat, especially in the children. There's several types of papillas which are located on the tongue. It is a filiform papilla, which are small and conical shaped. Situated over the dorsum of the tongue, this papilla contains less number of the taste buds. Fungiform papilla, which are round-shaped and are situated on the anterior surface of the tongue, near the tip. Numerous fungiform papillas are present. Each papilla contains moderate number of the taste buds. And circumvallate papilla, which contains many taste buds. They are large structure presented on the posterior part of the tongue and has in many numbers. These papilla are arranged in the shape of the V shape. Okay, so what is the main function of the taste buds? Taste buds contain about from 50 to 150 receptor cells and numerous supportive cells. Taste receptor cells have microvilli which extends through the pore, exposing the microvilli to the saliva on the surface, which visit the slows food molecules. Like olfactory cells, the cells of the taste buds are replaced continuously, each having a normal lifespan about 10 days. Substances called tastants dislodge in the saliva, enter the taste pores, and by various mechanisms cause the taste cells to deplorize. When the taste cells are going to deplorize, they release neurotransmitter that diffuse to the sensory neurons associated with the taste cells. The neurotransmitters stimulate action potential in the sensory neurons, which are conducted to the brain, where the sense of the taste is perceived. So, what is the main 5 primary taste? It is a salty, it is a sour, it is a sweet, it is the bitter, and umami. In this picture, you could see the localization of this taste. Bitter more located on the root of the tongue. Sour will be located on the lateral part of the tongue, the same about the salty, and sweet on the apex of the tongue. Umami will be located on the center of the tongue. The salty taste is going to be stimulated by the sodium ions. The sour taste is going to be stimulated by the acids. Sweets by the sugar and some other carbohydrates. Beta is going to be excitated by alkaloids, and umami by the amino acid glutamate and related compounds. The umami taste sensation is most intensive and coupled with a salty taste. This explains the popularity of the adding salt to the tomatoes, ketchup, soy sauce, and the food additive monosodium glutamate. Primary taste and unequally distribute over the surface of the tongue. Son salty, as we talked, proceed on the lateral part, sweet on the apex, and bitter on the root of the tongue. How the signals is transducted in the taste. So, salty will be caused by the presence of the sodium ions of the food. Due to this, it will be increased concentration of sodium ions responsible to flow it to inside of the cell. Voltage-gated calcium channels are going to open, and as a result, depolarization will occur. Calcium triggers neurotransmitters to release, which will stimulate the dendrites of the sensory neuron, which are responsible for the taste sensation. So caused by the presence of hydrogen ions in food. Hydrogen ions bind to potassium channel. Potassium channels will close. Taste receptors depolarize and calcium channels are going to be open. Calcium trigger release of the transmitters by exocytosis, and as a result, this neurotransmitter is going to influence on the dendrites of the sensory neurons, which transmit taste sensation. Beta associated with a variety of nitrogen-containing compounds. Also, beta molecules will block potassium ion channels. Okay, potassium is going to potassium leakage will decrease. Cell is going to depolarize and calcium channels are open. Calcium enters the cell and neurotransmitter is going to release. The neural pathway of the taste. Axon of the sensory neurons associated with the taste cell combine and contribute to the cranial nerve. Taste from the anterior part of the tongue of the 2/3 of the tongue is carried by the branch of the facial cranial nerve, which is called hornotympany. Taste from the posterior or 1/3 part of the tongue and superior of the pharynx is carried by the glossopharyngeal nerve. In addition to these two main nerves, the vagus carries a few fibers of the taste sensation from the epiglottis. This nerve extends from the taste buds to the nuclei of the medulla oblongata. Fibers from this nuclei decussate and extend to the thalamus. Neurons from the thalamus will project on the gustatory area of the insula and frontal cortex. Now let us move to the physiology of olfaction. Olfaction is sensation of the smell. It occurs in response to the airborne molecules called odorants entering the nasal cavity. A small superior part of the nasal cavity is lined with olfactory epithelium. 10 million olfactory neurons are present within the olfactory epithelium. Olfactory neurons are bipolar neurons with dendrites extending to the epithelium surface of the nasal cavity. The ends of the dendrites are modified into bulbous enlargements with a long specialized cilia called olfactory hairs, which lie on the thin mucous film in the epithelium surface. The mucus keeps the nasal epithelium moist, traps and dissolves molecules, and facilitates the removal of molecules and particles from the olfactory epithelium. So, odorant molecules bind to the molecular receptors and activate the G protein system. G protein will activate adenosine class. As a result, CAMP is formed, which binds and opens sodium, potassium, sodium and calcium voltage-gated ion channels, which will cause depolarization. Calcium also caused chlorine channels to open and chlorine to exit from the cell, increasing the depolarization. In the depolarization is great enough, action potential will be triggered. Information is going to spread through the olfactory cranial nerve. The axons from the synapses with the secondary ordered neurons in the olfactory bulb in structure called glomeruli. The secondary ordered neurons, mitral cells, form the olfactory tract, which goes to the olfactory tubercle. From the olfactory tubercle, olfactory information goes either to the olfactory cortex, concentrated with the discrimination of the smell, or to the limbic system, for triggering olfactory-driven behaviors such as sexual behavior. The primary olfactory neurons have the most exposed nerve endings of any neurons, and they are constantly being replaced. The entire olfactory epithelium, including the neurons, is lost about every 2 months as olfactory epithelium degenerates and is lost from the surface. Lost olfactory cells are replaced by a proliferation of the basal cells in the olfactory epithelium. This replacement of olfactory neurons is very unusual among neurons, most of which are permanent cells that have a very limited ability to replace. Thank you for your attention.